Cell organelles. Every cell has a membrane around it to keep its own contents separate from the external environment. We have seen this. Every cell has a membrane, skin-like structure. Why? What is the function? To keep its own contents separate from the external environment. Large and complex cells, including cells from multicellular organisms, need a lot of chemical activities to support their complicated structure and function. What do large and complex cells need? They need a lot of chemical activities. A lot of chemical activities take place to support. Why these chemical activities take place? To support their complicated structure and function. Since they are large, since they have complicated structure and they have many functions to do, chemical activities are needed to keep these chemical activities of different kinds separate from each other. These cells use membrane-bound little structures or organelles within themselves. So these cells have, what do they have? They have membrane-bound little structure. Again, little structures are there in these cells, and these structures are also membrane-bound. They are also bound within thin skin, and they are called organelles. And these activities are taken place. These different activities of different kinds are separated from each other because of these little structures called organelles. This is one of the features of the eukaryotic cells that distinguish them from prokaryotic cells. We saw prokaryotic cells do not have a proper nucleus. Eukaryotic cells have nucleus. So this is one of the important features of eukaryotic cells. Some of these organelles are visible only with an electron microscope. Now we are going to see what are these different organelles that help to keep the chemical activities separate from each other. We have talked about the nucleus in a previous section. Some important examples of cell organelles which we will discuss now are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria and plastids. They are very important because they carry out some very crucial functions in the cell. So let us see endoplasmic reticulum or ER. The endoplasmic reticulum ER is a large network of membrane bound tubes and sheets. Endoplasmic reticulum are nothing but they are tubes and sheets, and they are again membrane bound. They are covered, they are inside a thin skin. It looks like long tubules or round or oblong bags, vesicles. So how do they look? They look like tubes, sheets, tubules or oblong bags. The ER membrane is similar in structure to the plasma membrane. The membrane of endoplasmic reticulum is similar in structure to the structure of the plasma membrane. There are two types of ER, rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. RER looks rough under a microscope because it has particles called ribosomes attached to its surface. Rough endoplasmic reticulum is called rough because it has particles called ribosomes attached to its surface. On the RER there are particles, and these particles are called ribosomes. The ribosomes which are present in all active cells are the sites of protein manufacture. So we have to remember protein manufacture is done in the ribosomes. And where are ribosomes? They are on the ER, on the RER, on the surface of RER. The manufactured proteins are then sent to various places in the cell depending on need using the ER. So in the ribosomes, proteins are manufactured and then they are sent to various places in the cell, within the cell depending on the need. Wherever it is needed, it is sent. And again, using the ER, the SER helps in the manufacture of fat molecules or lipids important for cell function. Now we saw protein is manufactured in the RER. Now SER, smooth endoplasmic reticulum. Here what is manufactured? It helps in the manufacture of fat molecules or lipids. The fat molecules and lipids are important for cell function. Some of these proteins and lipids help in building the cell membrane. So now these proteins and lipids also help in building the cell membrane. This process is known as membrane biogenesis. So what is membrane biogenesis? Proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. Some other proteins and lipids function as enzymes and hormones. So see, these are the different functions of proteins and lipids: building the cell membrane and also as enzymes and hormones. Although the ER varies greatly in appearance in different cells, it always forms a network system. So ER is nothing but a network system. They are the tubes, they are the sheets, they form a network system. Thus, one function of ER is to serve as channels for the transport of materials, especially proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. So again we saw proteins and lipids, uses of proteins and lipids to form the cell membrane, and they also act as hormones and enzymes. Now we will see the function of ER. It is to serve as a channel. It is a medium. What is transported? Materials are transported, especially proteins, through ER. Where they are transported? Between various regions of the cytoplasm or between the cytoplasm and the nucleus. So ER is nothing but a network system. It is a channel, it is a medium for the transport of materials between various regions of the cytoplasm or between the cytoplasm and the nucleus. The ER also functions as a cytoplasmic framework, providing a surface. Now ER is also cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. The surface of ER serves as it only serves as a framework, cytoplasmic framework for biochemical activities of the cell. In the liver cells of the group of animals called vertebrates, SER plays a crucial role in detoxifying many poisons and drugs. Now in vertebrates, in the liver cells of vertebrates, SER, they play a crucial role. They function. What is the function of SER in liver cells of group of animals called vertebrates? To detoxify poisons and drugs. To detoxify means to make something, to remove the poisons and drugs, to remove the harmful poisons and drugs. Now let us see Golgi apparatus, another organelle. Just now we saw ER. There are two types of ER: endoplasmic reticulum, RER, rough endoplasmic reticulum, and smooth endoplasmic reticulum. We saw the functions of proteins and lipids. We saw the functions of ER. The Golgi apparatus, first described by Camillo Golgi, consists of a system of membrane-bound vesicles, flattened sacs arranged approximately parallel to each other in stacks called cisterns. Now, who described Golgi apparatus first? Camillo Golgi. And what are Golgi apparatus? It consists of a system of again membrane-bound vesicles. Vesicles are nothing but flat sacs arranged approximately parallel to each other in stacks called cisterns. Now they are in proper stacks, and these stacks are called cisterns. So these membranes often have connections with the membranes of ER and therefore constitute another portion of a complex cellular membrane system. So these membranes, they have connection with the membranes of ER, and therefore what do they form together? They constitute another portion of complex cellular membrane system. So see, membrane of ER and membranes of ER and these membranes of Golgi apparatus, they are connected. So these two membranes together form cellular membrane system. Like there is a network system, there is also membrane system. The material synthesized near the ER is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. Now what is Golgi apparatus? They are the sacs and they help in dispatching. That is, the material synthesized near the ER is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. So Golgi apparatus has two functions: packaging and dispatching of material that is synthesized near the ER. Packaging and dispatching the material that is synthesized near the ER inside and outside the cell is done through the Golgi apparatus to various targets. So its functions include the storage, modification and packaging of products in vesicles. What is stored? Products are stored in the vesicles, they are modified in the vesicles, they are packaged in the vesicles, in the bags. So what is the function of Golgi apparatus? Storage, modification, packaging of products in vesicles. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus. Also, one more function is making complex sugar from simple sugars is done in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. Also, Golgi apparatus is involved. The function is to form the lysosomes. So we saw Golgi apparatus described by Camillo Golgi. It is a system of membrane bound vesicles. They are arranged parallel to each other in stacks called cisterns. These membranes have connections with the membranes of ER and therefore they form complex cellular membrane system. Now the material that is synthesized near the ER. Now why ER membrane and Golgi membrane are connected? Because the function of Golgi apparatus is to pack and dispatch the material that is synthesized near the ER to various targets, to various places inside and outside the cell. This is one function. Second function, storage, modification, packaging of products in vesicles. Third function, making of complex sugars from simple sugars. That is also done in the Golgi apparatus. And number 4, formation of lysosomes. Now next organelle we will see, that is after ER and Golgi apparatus. Now we will see the third one, that is lysosomes. Structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. These enzymes are made by RER. Now, what are lysosomes? They are also membrane-bound. They are covered by thin skin-like structure. They are the bags, they are the sacs, and they are filled with digestive enzymes. What are lysosomes filled with? Lysosomes are organelles. They are filled with digestive enzymes, and these enzymes are made by the rough endoplasmic reticulum. Rough endoplasmic reticulum are those on the surface of RER we find ribosomes. Now lysosomes are a kind of waste disposal system of the cell. Lysosomes, the function of lysosomes is to dispose the waste of the cell. These help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. So now because of lysosomes help to keep the cell clean. How do they keep the cell clean? How do lysosomes keep the cell clean? By digesting foreign material as well as worn out cell organelles. Worn out cell organelles means the organelles which are now of no use. These two things are digested by the lysosomes. and because of that the cell remains clean. Which two things are digested by the lysosomes? Foreign material and worn out cell organelles. Foreign materials entering the cell such as bacteria or food, as well as old organelles end up in the lysosomes, which break complex substances into simpler substances. Now foreign materials entering the cell can be bacteria or food, and also old organelles. These both end up in the lysosomes, which break complex substances into simpler substances. So lysosomes break complex substances into simpler substances. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic materials. So we saw lysosomes are they are the bags filled with digestive enzymes. So they digest foreign material and old organelles which are of no use. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as the suicide bags of a cell. What happens during disturbance in cellular metabolism? For example, when the cell gets, what happens? The cell gets damaged. Suppose a particular cell is damaged, lysosomes burst. The sac filled with enzymes, it bursts and the enzymes come out of the bag, come out of the sac, and they digest their own cell. The whole cell is digested. It is broken down into simple substances, and therefore lysosomes are also known as suicide bags of a cell. Mitochondria are known as the powerhouses of the cell. Mitochondria have two membrane coverings. Now this organelle named mitochondria has how many membrane coverings? Two membrane coverings. And what are mitochondria? They are the powerhouses of the cell. The outer membrane is porous while the inner membrane is deeply folded. These folds increase surface area for ATP generating chemical reactions. Now how many membranes are there? For mitochondria there are two membranes. Outer is porous, it has holes, it has pores, while the inner membrane is deeply folded, it has folds. The membrane is folded all over, and because of this the surface area increases, and this surface area is used for ATP generating chemical reactions. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP molecules. So what is required? The energy is required for what? For various chemical activities. Why are chemical activities needed? For life, and this energy that is required for chemical activities is released by the mitochondria in the form of ATP, adenosine triphosphate molecules. ATP is known as the energy currency of the cell. ATP is also known as the energy currency. It is the energy of the cell. The body uses energy stored in ATP for making new chemical compounds and for mechanical work. So what are the uses of energy? What are the uses of energy stored in ATP? For making new chemical compounds. Either the energy is used to make new chemical compounds or for the mechanical work done. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. So, mitochondria why are they strange? Because they have their own DNA and ribosomes. Again, in mitochondria they are different. They have their own DNA and ribosomes. Therefore, they are really strange. Therefore, they are able to make some of their own proteins also. Plastids are present only in plant cells. There are two types of plastids: chromoplasts, colored plastids, and leucoplasts, white or colorless plastids. So plastids are present only in plant cells. They are not in animal cells. How many types of plastids are there? two. What are they? Chromoplasts and leucoplasts. Chromoplasts are colored plastids and leucoplasts are white or colorless plastids. Chromoplasts containing the pigment chlorophyll are known as chloroplasts. Now, what do chloroplasts contain? Chromoplasts contain the pigment chlorophyll, are known as chloroplasts. Chromoplasts containing the pigment of chlorophyll are known as chloroplasts. Chloroplasts are important for photosynthesis in plants. Chloroplasts also contain various yellow or orange pigments in addition to chlorophyll. So now, leucoplasts are primarily organelles in which materials such as starch, oils and protein granules are stored. So plastids are two types. One is chromoplast, they are the colored ones, and the other is leucoplast, they are white or colorless. So now, chromoplasts containing the pigment chlorophyll are known as chloroplasts. Now those chromoplasts that have chlorophyll in them, they are separately called as chloroplasts. Now why are chloroplasts important? They are important for photosynthesis in plants. Chloroplasts also contain various yellow or orange pigments in addition to chlorophyll. Other than chlorophyll, they contain yellow or orange pigments. Leucoplasts are primarily organelles in which materials such as starch, oils and protein granules are stored. What three things are stored in leucoplasts? Starch, oils and protein granules. The internal organization of the chloroplasts consists of numerous membrane layers embedded in a material called the stroma. Now, what is the internal organization of the chloroplast? How is chloroplast inside internally? How it is organized? It consists of numerous membrane layers embedded in a material called the stroma. There are membrane layers, they are layers of skin and they are embedded, they are placed in a material called the stroma. Everywhere there is stroma, and in that stroma these membrane layers are placed. These are similar to mitochondria in external structure. Like the mitochondria, plastids also have their own DNA and ribosomes. Vacuoles are storage sacs for solid or liquid contents. Vacuoles are again the bags, they are the storage sacs for solid or liquid contents. Vacuoles are small sized in animal cells, while plant cells have very large vacuoles. The size of vacuoles in animals is small, and in the plants, vacuoles are large in size. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. The size of the vacuole, the central vacuole in case of plant cells, is 50 to 90% of the cell volume. So vacuoles in plants are large in size. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. So in the case of plants, vacuoles are filled with cell sap and they provide turgidity and rigidity to the cell. Rigidity, they are not flexible. They keep the plants rigid. Many substances of importance in the life of the plant cell are stored in vacuoles. All the substances that are important in the life of the plant cell, they all are stored. These substances important in the life of plant cell are stored in the vacuoles. These include amino acids, sugars, various organic acids and some proteins. So what are these four things stored in the vacuoles? Amino acids, sugars, various organic acids and some proteins. In single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important roles in expelling excess water and some waste from the cell. So we saw in plant cells, vacuoles occupy 50 to 90% of the volume. They are big. Vacuoles in animals are small. In plant cells, vacuoles are full of cell sap They provide turgidity and rigidity to the cell. Many substances of importance in the life of plant cell are stored in the vacuoles. These substances are amino acids, sugars, various organic acids, and some proteins. In single-celled organisms like amoeba, what happens? The food vacuole contains the food items that the amoeba has consumed. The consumed food by the amoeba, the food that is consumed by the amoeba, is stored in the food vacuole. In some unicellular organisms, specialized vacuoles also play an important role. When in an organism there is only one cell, the organism is made up of one cell. In case of unicellular organisms, specialized vacuoles play important roles in expelling excess water and some waste from the cell. Vacuoles throw out excess water. They expel excess water and also some waste from the cell. Each cell thus acquires its structure and ability to function because of the organization of its membrane or organelles in specific ways. The structure and ability to function of a cell, it depends on the organization of the membrane, how the membrane and the organelles are organized in specific way. The cell thus has a basic structural organization. This helps the cells to perform functions like respiration, obtaining nutrition, and clearing of waste material or forming new proteins. So this helps the structural organization of the cell, helps the cell to perform different functions. What are the different functions? Respiration, obtaining nutrition, clearing of waste material, or forming of new proteins. Thus the cell is the fundamental structural unit of living organisms. It is also the basic unit of life. That is why, since all these important functions take place in the cell is very important, very fundamental structure unit of living organisms. Cell division. New cells are formed in organisms in order to grow, to replace old, dead and injured cells, and to form gametes required for production. New cells. Why are new cells formed in organisms? To grow, to replace old, dead and injured cells. Sometimes old, dead and injured cells need to be replaced. So new cells are formed, and also new cells are formed to form gametes that are required for production. The process by which new cells are made is called cell division. What is cell division? Cell division is the process by which new cells are made. There are two main types of cell division, mitosis and meiosis. So this cell division is very important because new cells are needed to grow, new cells are needed to replace old, dead and injured cells, and new cells are needed to form gametes required for reproduction. This cell division is of two types, mitosis and meiosis. The process of cell division by which most of the cells divide for growth is called mitosis. So cell division, when it takes place for growth, it is called mitosis. In this process, each cell called mother cell divides to form two identical daughter cells. Now in case of mitosis, each cell is divided into two same cells. They are called daughter cells. The daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissues and organisms. Now the daughter cells and the mother cell, they have what is same? The number of chromosomes is same, and this helps in the growth and repair of tissues in organisms. Specific cells of reproductive organs or tissues in animals and plants divide to form gametes. Specific cells of reproductive organs. Now there are some cells in the reproductive organs. They are the special cells or tissues in the animals and plants. There are special cells in the reproductive organs and tissues in animals and plants. These divide to form gametes, which after fertilization give rise to offspring. They divide by a different process called meiosis, which involves two consecutive divisions. Now in case of meiosis, the cells divide by a different process, and this process involves two consecutive divisions. When a cell divides by meiosis, it produces 4 new cells instead of just 2. In case of mitosis, a mother cell was divided into 2 daughter cells, but in case of meiosis, one cell divides, produces 4 new cells. The new cells only have half the number of chromosomes than that of the mother cells. Can you think as to why the chromosome number has reduced to half in daughter cells?